Bueno, buenas tardes. Vamos a empezar. Sí, vamos a empezar, por favor. Gracias.
Muchas gracias, doctora Ana Barahona, quien es el miembro del chair que representa hoy a la doctora, que va a presentar hoy a la doctora, a Dr. Evelyn Fox Keller. She will present. She's a professor from the Faculty of Sciences here in the National Autonomous University of Mexico. And welcome and thank you very much for being here.
No, thank you. Thank you. Well, the recent writing on the history and philosophy of science shows how it has been dramatically reshaped the concept of science by a new understanding of science itself. In the last few years, scientific knowledge has come to be seen as a product of human culture, an approach that has challenged the tradition of the history of science as a story of steady and autonomous progress. New topics have emerged in historical research, including the identity of the scientists, the importance of the laboratory, the roles of language and instruments, and the connections with other realms of culture and society, such as gender and rhetoric, fields in which Evelyn Fox Keller has done important contributions. In particular, on the history of genetics, a lot has been done by her and by others. In her insightful book, The Century of the Gene, Evelyn intends to show how gene talk has affected the course of biological research and points our need to examine the particular ways in which terms like gene action, genetic program, have participated in shaping the biological landscape of the scientists doing that work. The effects of such talk in theoretical as well as in experimental genetics is clear. The origin and application of new concepts have made it possible for genetics and evolution to be treated in an experimental way. I want to take a few minutes to talk about two aspects of this research that Evelyn has highlighted recently. The first is the notion of experimental revolution. New technical capabilities arise as a consequence of recombinant DNA. Our understanding of DNA increases our power to induce modifications in the gene sequences of many bacteria, plants, and animals. How can experimental practices be revolutionary? Research in this new field has a lot to say. The second aspect is rhetorical. Gene talk serves as a tool of persuasion. We see nowadays that gene talk is a common resource in popular thinking. It not only aids in funding and promoting interdisciplinary research projects in biology, evolution, or medicine, but also in creating bonds with the biotech industry. The closer such ties, the greater the research scientists' investment become in the rhetorical power of a language that works so well. Keller is insightful in showing how the obtaining of only limited results by the Human Genome Project is far from being a failure. It has reoriented the way we formulate our biological and philosophical inquiries. I have the pleasure and honor to present to you Evelyn Fox Keller, Emeritus Professor of History and Philosophy of Science at the Massachusetts Institute of Technology. She is a physicist and a historian and philosophy of science, and also a feminist. She is, Keller's early work concentrate at the intersection of physics and biology. And now her research has focused on the history and philosophy of modern biology and on gender and science. Welcome, Evelyn Fox Keller. Thank you.
They turn the light off here. Maybe we could put it on. The light.
The light. Si prenden la luz de aquí porque no se lee aquí, por favor.
Oh, thank you, Anna, for that fine introduction, and thank you to the organizers of this conference for inviting me. It's a great pleasure to come and actually have some time to visit Mexico. I've been here before, but it's always been in and out. The book that Anna tells you about, a little bit about, A Century of the Gene, was published in 2000, right after the first draft of the human genome sequence was published. And it was called The Century of the Gene. I mean, history, determined it. I mean, the sequence had been just published, and 1900 was the year Mendel's work was rediscovered, so I had a century. But the topic doesn't really belong just to that century. Of course not. I mean, I wanted to say that the book was a celebration, in my view. It was a celebration of the gene, of the power of that concept, but it was also an insistence on the conceptual nature of it, that it was a concept, and the question was whether it had now begun to outlive its usefulness, which I thought it did. The idea of the gene, the word gene had not been introduced, had not been coined until 2009. But the idea of the gene, of a particulate unit of inheritance, was much earlier. Not that much earlier, but it was 50 years earlier. And the second half of the 19th century was replete with arguments for some such notion. It was a brilliant idea that there was a particulate entity. For Weissmann, it was, let's see if I can do this now. Click here. No. Click here. One. Okay, so a frequent image for the importance of the gene, big. Is it big? It's big enough, yeah. It was the portrait gallery of the Habsburg lip. And it is remarkable. You see, it's far from a complete collection, but you see the portraits of the Habsburgs through the generations, and there is definitely a Habsburg lip. How do you account? Can't see it? I'm sorry.
Do you want to read that now?
Do I want to read that? Yeah.
Just because you need the lights on.
Oh, all right. Well, I want the light for the text, for my text. Okay. All right. You can't see it. I couldn't see the slides all morning. It's a casualty of the design, of the architecture. But the point is that these are very different faces on very different people. They look very different, but they all have the same lip. And the idea that you could account for that, it's okay. The idea that you could account for that constancy by hypothesizing an element, a particle that would persist, that would endure through the generations, unchanged, and that would, a particle responsible for that lip somehow, and it's in the endurance of the particle, the hardness of the particle, would account for the endurance of the trait. Now the notion of a particular unit of inheritance, as I say, came up many times in the latter part of the 19th century. For Mendel, there were their words were always different. Problem? No, don't worry. There are very few slides, and they're not important, and I'll explain them. Don't worry. For Darwin, there were gemmules. For Galton, there were stirps. For Weissmann, there were determinants. For De Vries, there were pangens. When Johansen actually formulated the term gene, he wrote, the word gene is completely free Can we turn this down? Is it echoing as badly for you as it is for me? Pardon me? I'm sorry.
Maybe it's bumping with the laptop.
It's something with the laptop? Okay, is this better? Okay. So Johansen wrote, the word is completely free from any hypotheses. It expresses only the evident fact that any characteristics of the organism that's specified in the gametes by determiners, which are present in unique, separate, and largely independent ways. In short, precisely what we wish to call genes. Now, built into the word and its precursors, maybe there were no hypotheses you could say, but certainly there were assumptions. The assumption that what accounted for that constancy of the Habsburg lip was a particle, an entity that itself, that the constancy of the entity was responsible for the constancy of the trade. That's not a truism. It was an assumption. It was an assumption that seems, once you thought of it, it was very compelling, very powerful, but it's an assumption nonetheless. Also, the assumption that these entities specify traits. Weizmann did not have the word gene, he used the word determinants, but like genes, they specified traits and they were effectively immortal. And because they specified traits, their immortality explains the endurance of traits. In particular, it explained the Habsburg lip. What is responsible for that Habsburg lip? It's a hard particulate entity, passed unchanged from one generation to the next. Now this duality that I want to focus on, a duality that in effect defined the particular unit of inheritance, led de Vries to suggest that what he called his name, the pangent, was part atom, like that fundamental element of the physical world, but a very special kind of atom. It was not only stable, not only endured, but it also had the ability to guide the development of a trade. It was more like a little organizer, a minuscule guide, a microscopic executive director, metaphors that are going to emerge over and over again. In other words, it was part atom and part organism. Now, of course, no one could say what a gene was or what it did, but of this dual capacity, its ability to endure and its power to direct, they were these two properties, they were absolutely confident. What is a gene? A particle that faithfully reproduces itself in every generation. What does it do? It acts. In the early days, its action is to make a trait. With that assumption came the further assumption that if a gene makes a trait, then the full complement of genes must make the organism. As Alfred Sturtevant put it in 1932, one of the central problems of biology is that of differentiation. How does an egg develop into a complex many-celled organism? That is, of course, the traditional major problem of embryology. But it also appears in genetics in the form of the question, how do genes produce their effects? Sturtevant continues, between the direct activity of the gene and the end product is a chain of reaction, and the task of the geneticist is to analyze these chains of reaction into their individual links. This way of talking, this view of the biological world is what I call, and the language and the imagery is what I call the discourse of gene action. Later, of course, we learn that what genes do is to make proteins. And the tacit view was that the full complement in the early days of the genes making proteins view, the tacit view was that the full complement of proteins made by the genome somehow makes the organism. We are the product of our genes. Accordingly, genetics, the science of heredity, was by definition the study of genes. In the early days, it was limited to the study of how these entities are passed on through the generations, to genes as units of inheritance. Now, everybody knows we inherit other things as well. We inherit cytoplasm from our mother's eggs. We inherit bits of RNA from both our parents, a taste for our mother's milk, for the food she gives us, our language, our names, our social world. If we're lucky, we might also inherit property, etc., etc. But for genetics, it is the inheritance of genes that count, just because they make us what we are. In 1934, Morgan put forth the critique that now in hindsight seems to have been so necessary. It seems so obvious. He wrote, "The implication in most genetic interpretations is that all the genes are acting all the time in the same way. This would leave unexplained why some cells of the embryo develop in one way, some in another, if the genes are the only agents in the results. An alternative view would be that different batteries of genes come into action as development proceeds, for which we had to wait another 30 years. But Morgan's critique was to little avail. What is obvious to us either was not obvious at the time to geneticists, or perhaps it just didn't seem important. This is the kind of thing that interests me. What is obvious and still ignored? And you see it in science all the time, over and over again. Later, with the advent of DNA, we learned to think of the gene as a protein coding sequence. And with this breakthrough, one might have thought the notion of gene action would have been laid to rest, but no. While the prominence of the image certainly declined, it did not disappear. Nor did the problem of regulation that so worried Morgan disappear. It did not even disappear with the introduction of Monod and Jacob's ostensible solution. In their model of the operon, regulation, they did introduce regulation, but regulation was achieved through the presence of another gene, the regulator gene. A gene coding not for a structural protein, but for a regulatory protein. One that acts by repressing the transcription of the original structural gene. As they themselves wrote, the discovery of regulator genes, quote, does not contradict the classical concept, unquote. Above all, it did not contradict, here we go again. I did nothing. Tell me if it gets to be a problem for you and we'll try to solve it, okay? Above all, it did not contradict the central dogma, nor did it contradict the early cartoon that went with it, DNA to RNA to proteins to us. Molecular biology made enormous strides in the decades that followed, adding enormous complications to the early picture with which it started. But even so, the simple framework provided by the discourse of gene action persisted to a degree I find rather surprising. Even though it was recognized as being clearly too simple, I suggest that it still nonetheless persisted, at least in some forms, until the end of the last century. One sign of this persistence, I think, can be seen in the story of junk DNA. Early on, molecular biology had introduced the definition of a gene as a protein coding sequence, and the definition stuck. Of course, it was soon confounded by many of the new discoveries, perhaps most dramatically by the discovery of split genes. But in practice, the early notion of the gene as a protein coding sequence showed surprising endurance. Indeed, for many, it served as a default definition of this increasingly difficult to define term. The preoccupation with genes thus readily translated into a preoccupation with protein coding sequences. In molecular biology and medical genetics and molecular evolutionary genetics, just about everywhere. Indeed, even after the discovery of vast amounts of extra DNA, DNA that had nothing to do with protein coding, this default definition of the gene persisted. Not too surprisingly, the vast amounts of non-genic DNA, repetitive, non-coding, nonsense DNA, that was found to reside inside genes, near genes, far from known genes, just about anywhere on the chromosome, soon came to be labeled junk DNA. Indeed, when Jim Watson first proposed the project to sequence the human genome, the proposal was criticized for just these reasons. Why bother sequencing all that junk? Bernie Davis, for example, referred to the plan as blind sequencing, complaining that, quote, it would be necessary to plow through 1 to 2 million junk bases before encountering an interesting sequence. Similarly, Robert Weinberg wondered how useful most of this information would be to anyone, since upwards of 95% of our genome contains sequence blocks that seem to carry little, if any, biological information. Well, as we know, the Human Genome Project was not stopped by these concerns about its wastefulness, about junk DNA. The project was launched in 1991, and by 2000, it began to release a number of pretty startling findings. Indeed, the Human Genome Project, which was never just intended for humans, Seymour Benzer quipped, the Human Genome Project is a project for learning more about Drosophila. The Human Genome Project caused another revolution, the genomic revolution, that may finally be shifting the framework that has guided us through the century of the gene. And I open by telling you that my book by that title was published in the year 2000, and that was before the genomic revolution. It was what unfolded, what unraveled through the course of the subsequent work in genomics was vastly different from what I and most other people anticipated at the time. Toño wants to tell you something. Toño. Perdón. Some of us are advising to have the mic here. We're going to try another method. There? Yeah. Well, is that better?
Oh, you can use it.
I can use it, whichever you...
I think that's much better.
Yeah?
Could you just...
Speak?
Yeah. Could you
Okay.
say
Yes. Is this better? Oh, good. Okay.
So we're going to start all over again?
No.
It's a joke.
The first surprise to come out of the Human Genome Project was the announcement of the small number of genes in the human genome, somewhere between 20,000 and 25,000, not very different from the number of genes in the lowly worm C. elegans. Two questions become obvious. What, if not the number of genes, accounts for the vast increase in complexity between C. elegans and Homo sapiens? The second and related discovery was of the very small amount of DNA devoted to protein coding, starting around 2% and eventually dropping to 1.2%. It's such a dramatic finding. You don't need to see this, but and what did they say I have to do? Yeah. So this is the part of the genome that's coding for proteins. This is the part of the genome that consists of genes. Got that? there's a lot, there's a lot else. With numbers like that, one has to ask, what is the rest of the DNA doing? Are we really justified in assuming that extragenic DNA makes no contribution to function? Well, what better way to find out than to look? In 2003, the research consortium ENCODE, Encyclopedia of DNA Elements, was formed with the explicit mandate of addressing this question. More specifically yet, ENCODE was charged with the task of identifying all the functional elements of the human genome. Never mind that we don't know how to define functional, but their heart was in the right place. What came out of this work was the realization that all that extra DNA was not just idly sitting on the chromosome, carried along from generation to generation without apparent function. Up to 80% of it is transcribed into into RNA. It may not be making proteins, but evidently it is making lots and lots of non-coding RNA. Far from idle, this wealth of non-coding RNA is busy forming networks of RNA and RNA protein components, tiny molecular machines that fill the cytoplasm and that, in interaction with incoming signals, seem to run the cell. They regulate not only the transcription of non-coding RNA, but also that of protein coding sequences. They also perform the work of alternative splicing, of epigenetic markings, of regulating the production and distribution of proteins, of managing the conformation of the DNA, and lots more. They're even implicated in the editing of other RNA transcripts and of modulating the configuration of regulatory networks these transcripts form. Such mechanisms seem to be especially active in learning processes, and I can show you another slide here, which shows the proportion of non-coding DNA as you move along, as the complexity of the organism increases. And you'll see where it gets to be close to 100% is in organisms with higher nervous systems, especially primates. Now, small wonder at this point that a new metaphor, something other than junk, came to be seen as required. Those vast regions of DNA that do not code for proteins soon came to be referred to as the dark matter of the genome. The place where, just as in cosmology, all the really important stuff happens. Again. The bottom line is that far from junk, non-coding DNA, the source of non-coding RNA, provides an immensely rich resource for the regulatory mechanisms that enable gene expression to respond to the outside world, especially to variations in both their proximate and distal environmental contexts. They provide the means by which organisms can adapt to changing environments. Most of the modifications they give rise to are short-term and more or less reversible, but they need not be. Those involved in development can persist for many generations of cell division. Some can persist through reproduction into the next generation. Unlike DNA, RNA sequences are malleable, eminently so, routinely rewritten and reinscribed. Through reverse transcriptions, such changes can also come to be incorporated in the DNA, turning the genome itself into what Jim Shapiro calls a rewrite system. But we mustn't forget that genomic sequences are in fact enormously stable. This stability is itself a consequence of an immensely complex system of editing and repair. And certainly, it's extremely important for understanding of biology. It may not be inviolable. This genome may indeed sometimes be rewritten, but it doesn't require that the critique that the genome is not stable, I think, is problematic and overused. Similarly, another issue under current debate is whether or not mutations are truly random, independent of their effect, as is virtually is always assumed in neo-Darwinism. A number of writers, including myself, have recently begun to question this assumption, but I don't think we should be too hasty. Many, maybe even most, changes in DNA sequences are indeed random mutations, independent of their effect. But the point I want to make is that they do not need to be. Indeed, in view of the ingenuity we're seeing in RNA dynamics, it would be surprising if evolution had not given rise to some sort of mechanism of directed mutation. But here, my point is to emphasize that nothing in the argument I'm making for reconceptualizing the genome as a reactive system depends on this. The mechanisms by which gene expression is regulated are quite sufficient by themselves to justify such a shift. Take, for example, epigenetic inheritance, the persistence of epigenetic markings across generations. It comes in at least two forms. First, through the transmission of such markings through the germline, and second, through the persistence of the behavior of environmental signals that induce these markings. Neither mechanism involves a change in DNA sequence, but both can eventually lead to changes in sequence by altering selection pressures operating on existing sequences. I'll get back to this later. But for now, let me return to ENCODE. The results of that research soon called forth a new, better, as I say, that dark matter, and I've already said that, so let me recapitulate now. What does it mean? And let me just summarize the argument that I seem to have elided from this text. The central point of my argument is that a shift is taking place in molecular biology from what I call the discourse of gene action to the view of the genome as a reactive system. And I attribute this shift primarily to the research coming out of genomics. But so now let me elaborate. What does it mean to say the genome is a reactive system? My claim is that it no longer makes sense to think of the genome either as a simple collection of genes or as a starting point, as the beginning of a causal chain that takes us from genotype to phenotype. Instead, we need to reconceptualize the genome, first of all, as itself a system, and second, as a reactive one at that. It is a subsystem of the cell that is composed of DNA that has been designed over the course of evolution to sense and to respond to the signals impinging on it. The sequence of the genome's DNA determines both the organism's sensitivity and its ability to respond to outside signals. It provides one of, if not the main mechanisms that enable organisms to adapt and endure. This it does through changes in conformation, chromatin remodeling, methylation of the DNA, in short, through many of the mechanisms associated with epigenetic inheritance. In other words, the reconceptualization or shift I'm advocating is dual. First, from the simple collection of genes to a digital script. In fact, not unlike Schrodinger's early notion of a code script, each part of which is in constant conversation both with its other parts and with the other constituents of the cell. And second, a shift from the simple causal chain from genotype to phenotype to a dense thicket of causal relations going every which way, connecting and interconnecting genomic sequences with the RNA molecules and proteins produced from it, with the networks these molecules form, and with the signals they both send and receive. In short, with all the parts and aspects of the complex system we now recognize the cell as being. Barbara McClintock was one of the first to anticipate such a reformulation. In 1984, in the closing remarks of her Nobel laureate speech, She described the genome as a highly sensitive organ of the cell that monitors genomic activities and corrects common errors, senses unusual and unexpected events, and responds to them, often by restructuring the genome. Here, once again, McClintock was ahead of her time. She anticipated a conceptual shift that was not yet apparent to most others, one that needed to wait for the establishment of the science of genomics and the lessons to be learned from its analysis before becoming evident to mainstream geneticists, and one that is closely akin to the shift to the notion of reactive genomes that I'm advocating. Why do I think this shift is so important? After all, aren't these just words? Action, reaction, junk, dark matter, genes, genomes. Why should words matter so much? Well, they matter to me. I mean, my preoccupation with words and language has really been one of the main themes of my professional life in the history and philosophy of science. My interest in words is I want to understand how they shape our mental landscape, how they highlight some features and obscure others. Take, for example, the discourse of gene action. That established a spatial and temporal map of development. Note, development begins with fertilization. There was no time. The notion of time before fertilization didn't exist. Maternal effects, traits initiated not by gene action but by factors, as we later came to understand, like messenger RNA, inherited from the maternal cytoplasm and not directly from her DNA. These are causal factors present prior to fertilization, but in the 30s, in classical genetics, they were renamed late effects and ignored. Nusslein Volhardt's great achievement was to light up a whole part of the campus that had been blacked out, and it opened up a whole new era of developmental molecular biology. I also could say something about code versus code script. We're going to cut that. Or take junk DNA. What does it matter if we call 99% of the DNA junk? Answer is, we don't have to pay attention to it. It matters for the uses of science too. Take, for example, medicine and our hunt for genes. Take the HapMap. Assuming that any SNP identified as being associated with a disease would lead to the identification of a gene causing that disease. That's the basic assumption of the HapMap project. But SNPs can be anywhere. Odds are overwhelming that they're not in protein coding sequences. The net effect is to ignore the role of DNA sequences that may be important in triggering the disease, but that are not associated with protein coding. The shift in language from junk DNA to dark matter brought non-coding DNA to attention, and has already opened up an entire new industry in medical genetics. I won't give examples. You all know the examples better than I do, or most of you do anyway. A similar story has begun to unfold about the role of non-coding RNAs in evolution. Traditionally, for reasons partly technical and partly conceptual, work of molecular evolutionary biologists has focused on changes in the protein coding sequences of DNA. But with the growing evidence of functionality in non-coding DNA, that's beginning to change. Of particular excitement have been studies of the role of changing gene expression patterns in the evolution of primate cognition. In a recent study comparing gene expression patterns in human, chimp, and macaque brains, Summel et al. Report that much of the divergence between species, what distinguishes cognitive development in humans from that of other primates, for example, are the changes in the expression patterns of genes regulated from afar by microRNA transcripts. Relatedly, in a 2012 review of the roles of non-coding RNAs in brain revolution, Qureshi and Mailer suggest that, quote, because the production of non-coding DNA sequence present in the genome correlates with organismal complexity, it is intriguing to speculate that non-coding RNAs preferentially mediated the accelerated and asymmetric evolution of the human central nervous system and underlie the unique functional repertoire of the human brain. Finally, Maddock and Mailer have documented the extensive role of non-coding RNAs in editing RNA transcripts in brain cells, thereby providing a means for cognitive adaptation to changing environmental conditions. More specifically, they suggest that such editing provides a mechanism by which environmentally induced changes in neural development and evolving brain architecture might subsequently be hardwired into the genome. Jump. One of the most interesting areas opened up by a focus on non-coding DNA is to be found in studies of biocultural evolution. Studies of the reactivity of the genome to signals beyond the cell, beyond the organism, to the world outside, and not just the external environment, but the world of other organisms, especially to the world of organisms with receptors for complex and long-range forms of perception, i.e. organisms with central nervous systems. Humans have especially sophisticated perceptual capacities, enabling them to respond to a wide range of complex visual, auditory, linguistic, and behavioral signals in their extended environment. And researchers have recently begun to show that responses to such signals can extend all the way down to the level of gene expression. Findings of this sort have led to a whole new wave of interest in the interactions between genes, genomes, and culture, and the multiple ways in which human culture has itself shaped the human genome. And not just human culture. Rat culture, rat behavior shapes maybe not the human genome, but certainly it shapes the rat genome. As, for example, when rats lick their pups, the licking induces epigenetic changes in the female offspring, which make them in turn lick their own pups, which ultimately change the selective pressure that leads to longer-term changes. This is a tremendously exciting time in biology, in good part because many long-held beliefs are being thrown into question. Dennis Noble goes so far as to claim that all the essential assumptions of modern synthesis have been disproved. Strong words, but Noble is far from alone in calling for a new look at neo-Darwinian theory. Indeed, a new extended synthesis of evolutionary biology. But while many echo his call for an extended synthesis, there's little consensus as to what that would mean. Debate prevails around a number of different issues. For example, much of the modern system was built around population genetics. In fact, it is in this framework that evolution is often defined in terms of changes in gene frequencies. Today, it may be clear to many that this is far too narrow a focus, but what is not clear is what role a modified calculus of population genetics might have in the new synthesis. For those motivated by recent studies of non-genetic forms of inheritance, the recommendation is fairly straightforward. Identify the entities exhibiting, if there are entities, exhibiting heritable variation, and extend the machinery of population genetics to accommodate the different levels of inheritance. I'm going to skip a little. This is a start, surely. That's just adding up all the variance that's coming from different mechanisms, different systems of inheritance. This is surely a start, but for one thing, it assumes a straightforward separation of genetic from non-genetic inheritance. Yet, such separation is far from straightforward. Similarly, the very meaning of genetic can be problematic, especially in relation to the notion of units. To simplify matters, let's restrict the term to that which is carried in the DNA. But what is to count as a unit of genetic inheritance? Any sequence or combination of sequences must surely count as genetic. It is inherited, it is replicated, and it mutates. But what is the unit? The list of possible sequences that can make a difference is long, even open-ended. And thanks to the availability of whole genome sequences, it's now possible to track all the genotypic changes of simple model organisms over the course of their evolution. But will this be describable in terms of population genetics? The full genotype to phenotype map also depends on the interaction between and among the genetic units, their products, and higher orders of inheritance. Extending the discussion to include multiple levels of selection is clearly progress, but it's also crucial to take into account the many kinds of feedback that operate through the system. For example, ecological variation feeds back on epigenetic variation, cultural variation on the selective advantage of genetic variance. The problem is not just that the interaction terms in these equations can be large, but also that each level of the system is linked to the other levels in complex causal loops between and among genes, epigenetic markings, ecological practices, niche variants, etc. One especially interesting new proposal is for the need to include feedback from the forces of the very force of natural selection onto the genesis of variants. But I, for one, do not see any way to do this within the conventional formalism. There are also other less controversial, even if not less difficult, effects that need including. For example, one of the uses to which non-coding DNA sequences are put is to regulate the activity of transposons, which in turn have a direct effect on mutation rates. Another is to regulate the states of methylation of the DNA, which in turn regulate gene expression. Both processes are, on the one hand, triggered in response to environmental signals, but on the other, made possible by sequence information in particular regions of non-coding DNA. That is, the changes in gene expression patterns induced by epigenetic markings, which in turn are induced by changes in environmental signals, are themselves mediated by non-coding sequences on the DNA. The challenges keep piling up. One especially hot topic these days is network evolution. The evolution of regulatory gene networks, metabolic networks, protein-protein networks, signaling networks, etc. Eric Davidson has taught us about the logic of cis-regulatory networks, and I very much look forward to hear his new thinking on this subject. But networks come in many kinds, and they exist on many levels, in many kinds of systems, and even in non-biological systems. And in all these systems, they undergo a kind of evolution. This more general problem, network evolution, has caught the attention of a burgeoning community of physicists, engineers, and systems biologists. Everyone agrees networks evolve, often very rapidly. But how? By what mechanism? If they don't themselves reproduce, they don't qualify as units of natural selection. What then is the role of natural selection in network evolution? And what else might drive the evolution of networks? Enter systems biology. Evolutionary biologists have long understood the importance of mutation and drift, but now they're being pressured to also take into account dynamics characteristic of complex systems, such as self-organization, self-assembly, self-organized criticality, blah, blah. Dynamics that give rise to the spontaneous emergence of stable properties in a single generation without the help of a transgenerational selection process linking the fate of stochastic variations to environmental pressures. For example, the physicist Barabasi and his colleagues had advocated a law of preferential attachment as a universal mechanism accounting for the ostensible ubiquity of what's called scale-free networks. I myself have been somewhat critical of this approach, in part because dynamics of this sort are characteristic of systems we might think of as having disorganized complexity, whereas the complexity of biological systems is so obviously, conspicuously organized. Organized into structures and architectures that themselves feed back onto their constituents. For the analysis of systems with organized complexity, engineers bring other approaches. John Doyle, for example, has argued for a different set of principles of network architecture deduced from dynamical control theory. Taking robustness to be the basic desideratum of biological organization, Doyle focuses on the fine-tuning that is required for robust control in specific contexts and on the inevitable trade-offs between robustness and fragility. Of particular interest is the prominence of what he and his colleagues call bow-tie architectures. Maybe I'll skip this about bow-tie, but they're a very interesting form of architecture, and the question is, why are they so common? How they've certainly been documented as shown to be highly efficient for the organization of development of properties we want biological systems to have. But how do they evolve? We know they get built into technological systems, but they also seem to be very common in biological systems. In the first part of my I think I'm going to skip this because I'm running late. Am I not? Yes. All right. What? Yeah. Okay. So, we owe Darwin the great metaphor of natural selection. It is a metaphor. And it's a metaphor that Darwin himself was aware of it being a metaphor. He wrote natural selection is analogous to artificial selection, a process by which animals and plants with traits considered desirable by human breeders are systematically favored for reproductions. Darwin may not have intended to personify nature, but the power of his metaphor was too great. Ever since, the literature on evolution has routinely referred to nature as the only active force in evolution. Nature scrutinizes, chooses, picks out, selects individual variants, while the organisms themselves play no role in either the genesis of variants or in their evolution. Indeed, between gene action and natural selection, the organism virtually disappears. The genotype generates the phenotype. Random genetic mutations are assumed responsible for the variation, and natural selection scrutinizes the range of phenotypic variants, selects those variants with success in leaving copies of themselves. In the new world, of reactive genomes that I'm trying to push, of epigenetics, of mobile transposable elements, of niche construction, of changing network architectures, of biocultural evolution, all that changes. Indeed, as these are all systems of inheritance that lead to differential fitness, they also lead to phenotypic resemblance between parents and offspring. But natural selection sees only phenotype. All right, forget that. Evolutionary biology has today become a theater with many more actors, not just nature. and these actors play many different roles, many of them at one and the same time products of evolutionary and developmental change, and at the same time themselves agents of change. Certainly organisms are far from the merely passive objects on which we used to think of selection as acting. They choose, the organisms choose, they modify, and indeed they create their own environments. Variation in structure or behavior, whether random or induced, genetic or not, leads to modification of these choices, often in doing so altering themselves by that very choice, altering the selective pressures operating on them. At the end of the 19th century, Weizmann had laid down the dictum that although the germline produced the soma could never affect the germline. The germline was the part of the organism that held its essence in perpetuity. In fact, for Weizmann, it was the inviolability of the germline that proved to him Lamarck had to be wrong. 70 years later, Francis Crick offered a rewording of Weizmann's dictum: information proceeds from nucleic acids to proteins and never back again. Another 45 years have now gone by, and we are obliged to admit that neither is strictly true, which is too bad, because the old picture made life so easy. Seeing genes, individuals, or even groups of individuals as targets of selection allowed for a clear mathematical apparatus for tracking selection. It was called population genetics. But now that we are obliged to replace all those one-way arrows in our causal diagrams by two-way arrows and cyclic loops, life, okay, I have a slide that, I just show you Weizmann on the central dogma that is due to, this one, it's due to Maynard Smith. It's a wonderful, so the top is the Weizmann picture and the bottom is the central dogma. But now we replace all those one-way arrows by loops and two-way arrows. Life has become extremely difficult. Selection is almost always multiple, operating less on individual parts and more on combination of parts that have turned out to be useful. Furthermore, these combinations produce their own selective pressures on the parts that make them up. Finally, selection may not be the only force that operates. In addition, it may also, one final word, become necessary to introduce time into our analytic framework. Survival, hence fitness, is cumulative, and if organisms are to count in the evolutionary game, they must persist at least until reproduction. In the real world, that means surviving over a course of both predictable and unpredictable changes in circumstances. Life is not static. Fitness, in other words, needs to be integrated over the vicissitudes of an organism's lifetime. In the real world, organisms must endure. They must be able to either resist or adapt to change, usually both. In a word, they must be robust. The questions I've been raising, and they've been all questions, no answers, are enormously difficult. But they are the questions that evolutionary biology now confronts us with. And they require our attention, and they are definitely hard. Certainly they're too hard, and I'm afraid too late for me, but not for you. For you, they constitute an extraordinary challenge and a wonderful opportunity. It's grist for your mill, and I envy you. Thank you.
We have about 10 minutes for questions. I'm going to speed things up a bit by trying to alternate the two sides, specifying a question from each side so that the microphone can be in the hands of the speaker when we're ready for the question. So, yes, we'll start with you with your hand up right there.
Well, thanks for your talk. And I just wanted to ask about what you mentioned of disorganized complexity in Barabasi's networks. Why are they disorganized? in your opinion, and as opposed to the networks that we see, for example, in gene regulation? Because Barabasi also is talking about social networks. So what would be the difference?
Barabasi is not, you could say Barabasi is not exactly talking about social networks or any particular, he's talking about models of networks. And the models are what's disorganized. The models are arrived at through statistical mechanics. Statistical mechanics is a machinery for eradicating structure. It's for getting rid of particularities. It's for averaging over all sorts of variables. And whatever organized complexity the system had to begin with, in the course of all of the statistical mechanical application, that organization disappears.
Question? Okay, way at the back, a hand waving. Stand up. And then I want to pick somebody from this side for the next question. The red arm up in the back. Oh, there was somebody right at the very back here who had their hand up. Changed your mind? Okay, we'll take this one here.
Well, you say something important that philosophy and technology and science have different visions, and for you, vocabulary is something important. But what is defined science now, right now? We have to let be guided by philosophers or by scientists?
Oh dear.
You say vocabulary is important, and vocabulary is something we all use. So
I think you need to listen to both. That's my own view. I mean, I think scientists, I think it's very valuable to have a kind of running commentary on what what scientists are telling you, a running critique. But I don't think, and I think there needs to be more dialogue between philosophers and scientists. But I wouldn't want to choose one over the other.
Okay. Question from this side. No? Question from this side?
See, I made your life harder by talking so long, but I make it easier by.
But I get to ask my question. This is not a deep philosophical question. Oh, sorry, right, I need to use the microphone. Is that okay?
No, try this one. Or try mine.
Who's in charge of the microphone? Hello? Oh, now it's on. Okay. The question is about the word gene. I've been arguing for ages that teaching genetics is only confused by using the word gene. But think about the Habsburg jaw, the idea that there is a particle that causes the Habsburg jaw, and it's a stable, durable, inheritable thing. Was there at the same time the concept of alleles, that there was also a particle for having a normal jaw, or a number of particles that caused different kinds of jaws?
Oh, it depends on who you're talking about. In some writings there was, yes.
Okay. Yes.
So in taking into account this vision of reactive genomes, would you say, or would you agree that around every different type of mutational event that we can imagine, there is this local biology around it, upstream and downstream, that can be quite complex and connected with many other elements, and that therefore it should be important to try to purify these mutational events as opposed to imposing them, for example, or just putting the mutations there, so that we can capture this whole story behind them.
Right. Well, but there are mutational events far from the gene that have a regulatory impact on the gene function. So it's this is a field, this is an area that's just beginning to get it unraveled. Does that answer your question?
Yes. Well, this would apply also to epigenetic mutational events and so on.
The epigenetical mutational events, it's hard to separate them out from the non-coding RNA, because the markings, the epigenetic markings, get translated into gene expression pretty directly, but whether which markings that get laid down and when and where are mediated by many different kinds of RNA machinery.
More questions? We have time for maybe one more. In the back, the gentleman in the gray sweater. Yes, you.
Hey, how do you think how biased do you think it's our view of complexity right now? Because we thought before that the number of genes would be the key to quantify complexity, and now we're saying that it's all the extra non-coding DNA. But I don't know if human is the most complex living creature there, and I don't know, maybe there are other mechanisms that also give flexibility to the genome that might not be considered when we talk about complexity.
Well, we don't have complexity is a big word that refers to a lot of different, and there are a lot of different ways of being complex, of organizing a complex system. The most primitive way is by having lots of parts. A slightly less primitive way is to having those parts organized in different, in lots of different ways in networks. You get more complexity by organizing all those different parts in hierarchical levels. There is no answer to your question. When people talk about complexity theory, it's a mistake. There is no theory of complexity. There are a number of theories of complex systems that are, virtually all of them have problems, but people who are interested in complexity are looking at different ways of conceptualizing complexity, of organizing it, of analyzing it. It's an open-ended field.
We're definitely out of time. So on to the next speaker. Thank you.
There's too much.
it's too loud, I can move it down. I can move it down. Do you want me to talk? Yeah. Hello. So if it's muffled, then I can move it down. I can slide it down. Yeah. Her voice was. All right. So it should be okay. If not, I could just move it.
No,
Okay.
no tenemos. ¿Tú traes este? ¿Traes para poner el micrófono? Es que no sabemos.
If it's muffled, I should move it further away. Her voice is muffled. You know, you can too loud over. We'll see how. Tell me if it's okay. Betty will tell me.
Okay. For this next session, it's my pleasure to introduce to you Dr. Otto Geiger. He belongs to the Ecological Genomics Program at the Center for Genomic Sciences of U.N.A.M.
When I start, we'll test.
So, thank you very much for the introduction. Berenice mentioned already where I'm coming from, so I can move on to the next slide. Can you hear me? Yep. Okay, so my name is Otto Geiger. I have the honor and the pleasure to introduce to you Dr. Bruce Alberts. Bruce has received his PhD from Harvard University, working with Paul Doty. He did a postdoc with Richard Epstein at the University of Geneva. Then he moved back to the United States, becoming an assistant professor at Princeton University, moving up the ranks to associate and full professor. Since 1976, he's now at the University of California, San Francisco. Since 1985, he's the chair of the department there. And, well, since 2013, he has a special title as the Chancellor's Leadership Chair in Biochemistry and Biophysics for Science and Education. I would shortly line out the research of Dr. Alberts. Well, he essentially got caught by the question, which was initially set up by this beautiful model presented in 1953 by Watson and Crick. How does this DNA replicate? And he dedicated much of his scientific lifetime in trying to resolve these machineries that are replicating DNA. And I think one of the surprises was that this was much more complex than anticipated initially. So these are really protein complexes which enable chromosomes replicate in as living cells divide. And he initially focused on a relatively simple system, the T4 bacteriophage, and resolved the individual contributing proteins in the DNA replication machinery. Later on, moving to chromosome organization, replication, and separation, often using Drosophila, but also other models. On his website, there's actually a very nice video conference where Bruce explains the replication fork when I began graduate school in 1961. Besides his enormous scientific achievements, Bruce also contributed enormously to the scientific community. He served as president of the National Academy of Sciences for 12 years. He was United States Science Envoy as of 2009. He was editor-in-chief of Science magazine for 4 years, and many, many more honors and awards. I will not list them here, but there are 16 honorary degrees, 25, he's on 25 advisory boards, and so on. I would like to mention here that when Bruce was editor-in-chief of Science, he wrote many insightful comments there, editorials, and I just would like to take this part, which is an excerpt of one of those editorials. And Bruce, after 5 years at Science, I came to realize that science is much more important for the world than most scientists suspect. The scientific values of honesty, respect for evidence, openness and tolerance are critical for every nation, and scientific approaches to problem solving are essential everywhere for meeting huge range of societal challenges. How true for this time and this country. Bruce probably is most known for his contribution as a writer. Since the original version of the molecular biology of the cell, he's author of that book, which is normally authored by multiple authors since 1983. It's now in its 6th edition, and Bruce always showed a very strong commitment to teaching, but also to science education of the general public. I would like to invite Bruce to give his lecture. I think it's titled Evolution and Biochemistry. Maybe it has changed, I don't know. So please go ahead.
Thank you for that wonderful introduction. We're testing the microphone. Is it okay? Can people hear? If not, raise your hand. It won't work. I was very good to follow Evelyn's very nice talk. I mean, writing a cell biology book every 5 years, you sort of get a sense of how our view of life, the molecular basis of life, is changing. And the main striking thing I would say, and I'll emphasize this in my talk, is that every 5 years when we think about it carefully to write a new edition, we realize how naive we were before about the chemistry of life. Every 5 years it gets ever more complicated. and the kind of issues that Ellen talks about reflect the sophistication of the chemistry. And the main thing I would say to young people is we're far from understanding even simple cells, so there's a huge amount to do. We need you all to be skilled scientific researchers. So I'm going to start with a little personal history because there are many students here and they love to hear this little bit, and you'll see why in a second. I was in high school when this famous picture of Watson Crick and their discovery of the DNA double helix was taken. Watson was only 25 years old, a reason to believe in young scientists, another problem these days. And I would especially like to congratulate all of you for letting your students organize this conference. We need to do much more to empower young people everywhere around the world. So Watson and Crick, with this beautiful structure and a one-word sentence at the end of their paper about how this would explain heredity, they had really solved the problem, and they were right. But they didn't know anything about the machinery that made it possible to copy DNA from one generation to the next. And starting as a graduate student in 1961, I would spend 30 years working on this problem. It was a great mystery to solve, and that's a lot of the fun of science, solving mysteries. And they actually pay you to solve mysteries. This is amazing. Uh-oh, now we're in trouble. Oh, I hit the wrong button. Okay. I did something terrible. I hit a... Uh-oh. Let's see. There are too many buttons on this machine. Let me see. I somehow turned it off. I hit that screen button. Maybe you could oh, I see what happened. Oh, I see what happened. It stopped the whole thing. Okay, let's start again. Okay. Is that working now? Yeah. Okay. They have too many buttons on this controller. I'll try not to do that again. Okay. So it's important to talk about how we viewed this cell when I was a graduate student. I was in a physical chemistry lab. Physical chemists know the amazing rate at which molecules collide. We therefore thought of a cell as a tiny test tube full of very concentrated macromolecules and small molecules, all freely diffusing in that space, not organized in any special way. So when we thought about DNA replication, the problem I was working on, we thought that it would work like this. Now, after Watson and Crick, in 1957, Arthur Kornberg discovered DNA polymerase. That also earned him a Nobel Prize. But it was thought when I started that this one enzyme replicated DNA, and that the way it would work is that it would randomly collide with a growing end of a DNA chain, add one nucleotide using the template base pairs to transfer the information, fall off, then a new molecule of DNA polymerase would collide with the template, and so on. That's the way all of chemistry was supposed to work. This is the good part for the students. I had a very dismal career as a graduate student. I really didn't know how to do science, but I thought I did. But like many others, we were inspired by Watson and Crick's successful prediction. So I made a theory about how DNA polymerase by itself could replicate the DNA double helix, even though in the test tube we knew that DNA polymerase would only work on DNA once the strands had been separated. I did many experiments to try to see if the theory was right, and of course it wasn't, and I got many no answers. Nevertheless, my professor, after 5 years, said, write up a thesis, you've done enough, you're going to graduate. This is at Harvard University. The last thing you do there is you walk in the room with famous professors and they talk to you for an hour or half hour, depends on how busy they are, and they pass you and you go off. So my wife Betty and I had given up our apartment. We had tickets next week and a 1-year-old child to take to Geneva, Switzerland for my postdoctoral fellowship, and nobody ever failed at this stage, but I did. And this was one of the most formative experiences in my career, looking back. And in general, I would say that those people who are successful have failed a lot and learned from their failures. I've never learned anything from my successes, only from my failures. And this is why older people can be useful sometimes as mentors. We've learned a lot from our failures. So this is a little write-up of what I learned. If you want to know more about this, there's a wonderful website put up by my colleague Ron Vale at UCSF called iBiology. It's got hundreds of scientific talks, but also has this little section on called iBio Magazine. I gave a little short talk there on the importance of failure. Uh-oh, I did it again. Now this is a big test. How many times do I have to fail before I learn? When I was younger, it was only once, but as you get older, it gets slower. Okay. So what did we learn? We learned that theoretical biology is much more difficult than we had expected. But most importantly, I spent a month just thinking whether I could do science or not before I did anything. And I came to realize that having a good strategy in science is the secret to success, and my strategy was terrible. And there are millions of experiments you can do, most of which you shouldn't do, so you have to think very carefully about how you spend your life as a scientist. Okay, then I moved to Geneva, finally after 6 more months of experiments, and my wife and I were homeless for 6 months with a 1-year-old child, but anyway, we finally did pass, and I went to Geneva, and almost immediately I discovered that my whole hypothesis was wrong about DNA polymerase being the only enzyme needed to replicate DNA. And the reason was I discovered this wonderful model organism. Max Delbruck, a German physicist, came to the United States, was a leader in establishing the early days of molecular biology. This was one of the main organisms used. And there have been a lot of genetics done on this bacteriophage. And what I hadn't done was read the literature in genetics. I was a biochemist. I only read biochemistry. And in the middle of my thesis, the group at Caltech had published genetic analysis of this bacteriophage that proved that many different proteins were needed to replicate DNA. In fact, 7. These 7 different proteins, those genes were given the numbers because nobody knew what they did, 32, 41, 43, so on. But one of these was the gene 43 product had been shown by others to be the T4 bacteriophage DNA polymerase. So clearly, at least 6 more proteins were needed, and how then to proceed? And basically, what we did was use the genetics to find the proteins and then try to do the biochemistry. Finally, it did work, and we discovered that these proteins fell into different groups: the DNA polymerase holoenzyme, so-called single-strand bonding protein, and so on, and that, most importantly, they worked as a protein machine. Of course, this was not only my work, it was the work of many laboratories working in different systems, but basically we came to recognize that DNA is made in a very sophisticated way. There are 2 DNA polymerase enzymes at any time working on DNA. One has to go backwards. The one on the bottom here could go forward on the leading strand. It does so very rapidly, 500 nucleotides per second. But the other one has to go backwards because the polymerase only goes one way, yet the 2 strands are anti-parallel, as Watson and Crick had shown in their 1953 paper. So this one on the lagging strand has to make DNA backwards, a series of short fragments, and this involves forming a loop. The reason why I'm showing you all this is because I want to show you my favorite movie, which is a movie of how this all works. And this process I'm showing you not because it's DNA replication, but because this is how all of life chemistry works. It shows you how sophisticated the chemistry of life is. And we're lucky that that's true. Otherwise, I couldn't be talking to you and you couldn't be listening. Oh, I did. Okay. So, this is the DNA moving at the actual rate it moves inside billions of your cells right as you're sitting here. And here it goes. Actually, there's an audio with this, but I didn't plug that in. So at the bottom, the DNA double helix is coming in. It's been separating by this DNA helicase that used ATP energy to open the helix. This is the leading strand. The enzyme just going continuously stays in place. It never falls off. Goes for millions of nucleotides. And the other side, the other side is where it gets all complicated. This is really the nature of life. This little thing right there. This is how everything works. So the other polymerase has to be used repeatedly, and the DNA is drawn out in loops to make these short fragments that are eventually sealed together, so-called Okazaki fragments. And if anybody had told us that this was the way DNA would be made, before it was discovered, we would sit there crazy. This has to be made with DNA is made with one error in a billion base pairs. How could you make DNA in this way with all these little short fragments and all, get them all sewn together with no mistakes? Well, that's the amazing chemistry of life, and we still don't fully understand all of it by any means. Okay, so we now know that the same basic mechanisms used to replicate DNA from large viruses like T4 bacteriophage, large DNA virus, to mammals like ourselves. But as evolution proceeded, you add on more and more things. I mean, each protein in that system has bells and whistles added to it, subunits that are regulatory, many things. It gets more and more complicated. Just a few weeks ago, this breakthrough paper was published from the UK, John Diffley's laboratory. It's published in Nature. Basically, they have reconstituted the eukaryotic replication proteins. I only had to use 7 different proteins. They have 16 purified replication proteins made from 42 polypeptides. So that's how much more complicated it gets. And here's a diagram from their paper outlining. I don't expect you to actually see this. You can see it's complicated, but it's elegant, and it has to be elegant in order for us to be alive. And this is only the tip of the iceberg, because this involves maybe a tenth of all the proteins critical to DNA replication and DNA repair. So there's a huge amount left to do. But by reconstructing these processes in a test tube as they've done, we could now get to the molecular details. And we now know that basically almost everything in the cell is driven by a complex of 10 or more proteins that work as what we call protein machines. These protein machines function very much like the machines in everyday life that are driven by electric energy. They also need energy because they have to move unidirectionally. Their ordered movements are driven by proteins in the complex that harness the energy of tightly bound ATP or GTP molecules to drive unidirectional allosteric shape changes in the protein. And you were seeing some of that in that last movie. That of the 7 proteins, there are 3 of them that hydrolyze nucleoside triphosphate. Okay, so an important challenge for the next generation of cell biologists is to obtain the information needed to accurately describe the mechanism of every type of protein machine in a cell. This will require the reconstitution, purification, biochemistry of hundreds of protein machines from their purified components, so that the detailed chemistry of each machine can be deciphered through reactions studied in a test tube. Let me just say that after 1975, we finally had all those proteins, 7 proteins. We mixed them in a test tube. We had this wonderful day where all of a sudden we could replicate double-stranded DNA. But it took us another 6 years to figure out what was going on. And that we could only do that because we could change all the proteins and manipulate the system. You can't do that without purified system. Okay, so understanding the molecular details of these processes will be critical to improving human health. For example, cancer selects early on in general for increased instability to generate all those mutations you need for tumor cells to progress, to be dangerous. And there's a great potential in exploiting each particular defect to eradicate cancer in some form of personalized medicine. And in fact, the new head of the UCSF Cancer Center is Alan Ashworth, who just moved there this January. And he developed, one of the developers of the so-called PARP inhibitors, a FDA-approved therapy that kills tumor cells of certain classes specifically by exploiting their defects in DNA repair. Okay, so next, learning from textbook writing. These are the first 4 editions. This first edition, it was Jim Watson's idea. Jim Watson's a brilliant guy, but he told us all it would only take 2 months to do this. A month in the first summer and a month in the second summer. That's how he got us all to do this. In the end, that was off by a factor of 7. So we spent a huge amount of time, and it was almost another failure. I described that in my talk. This is the original set of authors. That's a much older Jim Watson at the back there, crossing Abbey Road. And the reason why we're crossing Abbey Road is that basically most of the book was written about 2 blocks away from there, with us all sitting in the same room 7 days a week, 12 hours a day. Oh, I did it. Okay. This is the 6th edition, and I already told you, every time we write, we realize how much we don't know. I'm very proud of this time. We finally admitted that. Students get the wrong impression that, we write in a textbook what we do know, and they think that we know 95%. I can assure you we know 5%. So at the end of every, the most fun of this edition was preparing what is it we don't know that's really important, and these are at the end of each chapter. So I'm going to just tell you a bit about what I learned, what we all learned, that was surprising that we didn't realize previously. First of all, the recognition that extensive sets of scaffolds, special protein and RNA molecules, produce biochemical subcompartments in the cell without requiring a membrane, and therefore the cell is nothing like a test tube, nothing like the way we thought. Everything inside of a cell is organized. The chemistry is organized and compartmentalized. And secondly, the recognition that positive and negative feedback loops underlie nearly all cell chemistry. Evan mentioned this, creating complex networks of interactions that give rise to so-called emergent properties that will require new computational methods to decipher. And I'll talk about both of those. So let's talk first about scaffold proteins. This is a diagram from our textbook that just illustrates why you have scaffolds and why that's so important for cell chemistry. You concentrate a bunch of reactants in a small space. Usually these scaffolds are located in a specific part of the cell, but also they concentrate all the reactants, so the reactants give products much more efficiently and rapidly than they otherwise would. And that's obviously important for life to exist. This is a very simple scaffold that was discovered by Tom Pollard's lab to help actin polymerize more rapidly than it would otherwise. They found when they're doing the biochemistry that the actin polymer would polymerize, catalyzed by this protein formin that binds to the growing end. It would polymerize at a rate that's faster than so-called diffusion controlled. That means it polymerizes faster than actin could ever collide with the end normally. But the reason that is possible is that there's a scaffold on the formin molecule. This yellow molecule, this whisker carries actin monomers loaded, ready to hit the end, and so that greatly speeds up the rate of actin polymerization. That's probably the simplest case of a scaffold I know about. Much more complicated scaffolds are in cells. These have been the ones that are involved in protein kinase cascades have been well studied by Wendell Lim at UCSF and others. Scaffolds are involved in forming many biochemical factories inside the cell. This is a picture of a stained cell nucleus. Can't really see the red, but there's separate aggregates of proteins involved in transcription, and the green ones you can see. Those are the aggregates of the proteins involved in DNA replication. So the reactions are faster because everything is concentrated in specific places, making separate biochemistry compartments within the cell nucleus in this case. And what life is really like is represented by this cartoon. This is the way we draw things when we don't understand them. The kind of cartoons that we have in our textbook basically are at the end of every paper now, but they're misleading. They suggest that we understand something when we don't. But this is the complex of proteins that underlies synapses. There's a special chemistry on both sides of a synapse. Special biochemistry because of these scaffold proteins on each side that hold special proteins there. Otherwise, we couldn't talk, think, or do anything else. And what we need to do, of course, is do the kind of biochemistry that's been done on DNA replication and on protein synthesis to really see how these compartments work and hardly anybody's working on it. So another wonderful project for young people. And I just, in the context of the talk this morning, think about an RNA world. Well, we know that RNA scaffolds, these long non-coding RNAs, thousands of them, many of them are really functioning as scaffolds, holding other reactants together to do multiple different things in the cell. And it seems pretty sure to predict that in an RNA world, these RNA scaffolds were also very critical for making the biochemistry work. You could get confined reactions without even having a membrane. Of course, we don't know anything about that, but again, something that we hope to find out in future years. So what about emergent properties? Well, they're all over the place. This is how nearly all of biology works. When I was a student, we said A goes to B, goes to C, goes to D, goes to E. And that's, of course, right. But while they're doing it, there's all these feed forward and feed backwards loops that make this process useful for the cell, so it can be regulated. And there's no way to understand such pathways without mathematics. I'm going to give you a simple example of this kind of situation of an emergent property. Well, actin is a major cytoskeleton protein inside of the cell. It forms a filament. Here's a filament right here. This is just in a yeast cell. These are all the actin-binding proteins that it interacts with. We can't really make sense of this with the human mind. Once you get beyond a few proteins interacting, it's just too complicated. We need to use new kinds of methods. And even when we have a complete knowledge of all the molecular molecules, protein machines, and molecular interactions in a cell, we will not be able to understand it. Instead, life reflects the emergent properties that result from very complex networks of interactions. So just collecting data, keep on making catalogs of all the proteins in the cell, all the RNA molecules in the cell, this is not understanding. It's not sufficient for understanding. We will need mathematical or computational models to decipher these complex systems, and I'm going to just give you one example. Because, everything, as I've said, is about spatial organization inside of a cell. So we not only need mathematical computational models, we need models that can deal with spatial parameters in 3 dimensions. So modern computers make possible individual molecule-based simulations, so-called agent-based modeling, in which tens of thousands of molecules are simultaneously allowed to diffuse randomly and react in a virtual space. Thanks to modern computers, you can run many billions of such cycles and simulate the positions of all the molecules in cycle steps of micro to milliseconds. And after running the computer for 10 hours or so, you see what happens to those molecules. And if those molecules give you a property like the actin cytoskeleton doing what it does in real life, then you can start to think that you're really beginning to understand it, and more importantly, you can predict the experiments to do that will test whether you really understand it. So in order to do this, you need a large amount of quantitative data. Otherwise, there's too many parameters, and the modeling is not useful. So you need to know all the molecules, you need their partners, you need rate constants, you need to do on and off rates, and you need to be able to compare what you're doing in the computer with what happens in the test tube or inside the real cell. And so one of the early models selected, it has to be pretty simple, was the movement of the Listeria bacterium inside a cell as a model for the actin-mediated movement of intracellular vesicles. And the reason why this happens is that the bacteria hijacks the normal cellular system when it comes into the cell, causes the host cell to polymerize actin filaments on its tail, push it around the cell, eventually push it from cell to cell, which helps it avoid the immune reactions. And the reason why this was useful is that an in vitro system had been worked out in France in which a mixture of 5 purified proteins moved a polystyrene bead mimic of the bacterium. And secondly, nearly all the rate constant affinities for these 5 proteins in the set were determined. And so this enabled the modelers to actually have enough constraints to see whether this process that had been set up actually was understood. And here's the actual bacterium moving on the left. Let's see. Oops, back. Okay, I got to master this thing. And the model on the right, you see that, I'll go back again. The two are moving at the same rate and the same kind of motion. Next slide shows a higher magnification view of this. You really can't see the colors. Yellow and red the red's disappeared. But basically, you could get a feeling that we actually understand this process, how actin polymerization using these 5 proteins is moving this cell. So now you have to go build this up to a more complicated system, and Tom Pollard at Yale has been working on trying to understand another actin-based system, how the contractile ring that drives the separation of one cell into 2 in cytokinesis, how that works. And he's using a model system. He's using fission yeast, where he has all the genetics and proteins and a lot of advantages over using a mammalian cell. And he's actually able to learn a lot by using modeling, comparing it to experiment, and going back and forth between modeling and experiments to convince him and hopefully others that we really understand a lot about this process now. The next slide is one of his slides. You just lent it to me. This shows some of the modeling that they're doing, because what happens is the actin are in these nodes containing actin and multiple other proteins. The nodes come together to form a ring, and then the ring contracts. And the ring will self-assemble in the model, and the ring tension that you could get from the model matches the tension that they could actually measure. So this is the future. In my view, this is the future of understanding really how cells work. And there's a heck of a lot that need to be done. Close coordination between skilled computational mathematical thinkers to make new methods and experimental biologists. So my conclusion, it will probably take most of the century to gain a true understanding of how cells and organisms work. We're going to need much more biochemistry and purified systems that reconstitute biological systems, like the example I gave you from for the initiation of eukaryotic replication, the example of moving a polystyrene bead with actin to mimic that process. But we're also going to need new quantitative methods for analyzing and understanding the enormous complexity of life's chemistry. The methods we have are allowing us to make progress, but they're certainly not up to the task of handling the enormous complexities. So now, to get to the topic of this meeting, how do we make use of evolution to help us with this? So my conclusion is that, again, nearly all cell processes will be based on elegant mechanisms too hard to predict, but there are remarkable homologies between living things. We saw that already between my bacteriophage and how human DNA replicates. And just as in that case, a shortcut to understanding humans would use model organisms wherever possible. This is the yeast Pombi that Tom Pollard's lab is using for cytokinesis. This is the other yeast, or VCI, used for many important research projects. Famous fruit fly, and of course I could have many others here. But the major point would be that we ourselves, mammalian cells, are so complicated, we need help from simpler systems. And because evolution occurred, these systems are remarkably homologous. Unfortunately, this is my political statement. We in the United States have been overemphasizing translational biomedical research when basic research on biological mechanisms remains absolutely crucial for improving human health. And the reason is that neither politicians or disease advocates understand the kind of things I'm talking about. They don't understand that things are so complicated that we need a lot more science. This is my favorite editorial from my 5 years at Science. It was not written by me. It was written by a wonderful translational research, MD, Huda Zogby, who is famous for studying Rett syndrome, neuroscience. She called it the basics of translation. She had a special insight due to the fact that she grew up in Lebanon, so her native language was not English. So translation between English and other languages was her analogy. And what she basically said is at the top. The challenge in translational medicine is that scientists are trying to translate a text with the sophistication and depth of Shakespeare using a first grader's vocabulary and experience. That's such a wonderful insight, and it's something that everybody should be able to understand, even if you don't know any science. One example, again, come back to Drosophila. We're still far from understanding how cells work together to form and maintain tissues. We got a huge number of examples that working out mechanisms in Drosophila provides a shortcut to understanding humans. We wouldn't almost know anything about human development without the early work in Drosophila genetics that revealed the important proteins involved. And in fact, today, in our last edition, the chapter on the development of tissues contains 50 references to Drosophila, which is 4 times more references than to the next most cited organism, which was the mouse. So it's not only that it was useful, it's still enormously useful. Consider the human brain. If you don't understand what emergent property is, think of human consciousness. Human consciousness comes out of a network of nerve interactions. So that's the ultimate emergent property. Here's a little bunch of stuff from our textbook. Look at this, how complicated the brain is. It's made out of neurons, of course. Very complicated cells that look like this. The human brain contains more than 100 billion neurons, each of which on average has to make connections with 1,000 others according to a regular and predictable wiring plan. And you get, this is a little slice from Golgi of a human brain, or at least a mammalian brain. So we need help. We need to work with something simpler. And here's the human, here's something simpler. The Drosophila brain, it's only 1 millimeter across. It has 100,000 neurons, 1 million times less than humans. We got all the genetics, we got all the tools, we can manipulate the fly. We can't do experiments on humans. There's wonderful work being done to reconstruct the wiring networks of, so far, over 16,000 single neurons have been reconstructed in the Drosophila brain. This is just a brain with 7 of the 100,000 total neurons in it. So basically, we're never going to understand the human brain until we understand the Drosophila brain. That's what I would say. And yet, people want most of our young people in the United States to work on mammalian systems. So this is a sobering paper just published in the journal Genetics entitled Fruit Flies in Biomedical Research. And they point out that the mutant phenotypes for thousands of fly genes can be corrected with human homologs for a cDNA encoding a human protein. So the evolution has made us so similar to flies, worms, even yeast, that this is obviously the way to figure out how humans work. And yet, there's been a 30% drop in our National Institutes of Health U.S. support for Drosophila research in the past 5 years, when the overall drop in support for research has been 15%. So over and over, people are making mistakes about what's really important for the future in biological research. So we also need many new models. We have maybe 7 classical model organisms. I give a lot of credit to a young faculty member at UCSF, Wallace Marshall, who had the guts to go back to an organism that T.H. Morgan worked with, Stentor. Here's something you think about we not understanding. Stentor is a giant single cell, and like it's a ciliate, And yet it's got all these structures. It's got, a mouth, it's got food vacuole, all kinds of organized patterns on the surface. And T.H. Morgan and then later Vance Tarter and others showed that you could cut this single cell up in little pieces, paste it back together, and it would regenerate its structures. So I would claim that we really don't understand much about the cytoplasm even. So as Wallace says, and this is his first paper he published, the principles of morphogenesis and pattern formation in single cells remains largely unknown. Although all cells develop patterns, they are most obvious in ciliates. Hence we have turned to a classical unicellular model system, the giant ciliate stentor. We also need new technologies. You all probably know that 99%, once we started sequencing DNA in soil and in the ocean, we realized that 99% of the organisms there we can't grow. So you talk about dark matter. There's 99% of the life on Earth. It's dark matter. This is a new technology called the iChip, where they basically make a sandwich with many wells of single cells taken from the environment. They then show that instead of not being able to grow 99%, now they can grow about half of all cells by putting this back in the environment from which it came. And this is a way you can clone, characterize, and sequence organisms, huge numbers of organisms we don't know anything about. So here's my dream. And we don't know how to do this. Can we combine a new ability to culture organisms in situ with new robotic single-cell sequencing techniques to focus on discovering valuable missing links in the 99% of all organisms that could previously not be grown in laboratories? Just to take a random example, there's this very large, complicated protein complex called the intraflagellar transport protein complex that allows cilia to grow and be formed. We don't know where this thing came from. We don't have organisms with intermediate stages. In this and many other cases, if we could find ancestral versions and work out how they work, this will be important for guiding experiments on their more elaborate later relatives. So what I'm saying is, can we work out a way to look specifically for organisms in the soil, in the ocean, or somewhere that would have more primitive versions of this complex, or more primitive version of some other complex some other function, many, many different ones, rather than just having to isolate everything and then looking later. So that's a real challenge, and of course it would involve a lot of robotics, a lot of sophisticated screening technology. But anyway, this would be one way to use evolution to find shortcuts to understand all of life on Earth. Okay, so my last slides. In summary, where has biology been? In the past 50 years, tremendous advances have been made in our understanding of the molecular basis of life, driven largely by the development of powerful new techniques. Genome sequencing is only one of them. We get 1 million times cheaper to sequence DNA than it was 20 years ago. So for well-studied organisms, we could see our way to the end of remarkable descriptive phase in cell biology. If anybody told me in 1960 that we know about all the proteins in any kind of a cell and the gene sequences, we wouldn't have believed them about that. But we could do that. We need, a lot more work to do. We could get all the descriptive, we could get their structures, but still, the problem remains. Where is biology going? We now know that the chemistry of life is incredibly complex, by far the most sophisticated chemistry known. Many of the most interesting attributes of life are due to emergent properties that stem from very complicated networks of chemical interactions whose consequences cannot be deciphered from the details of a few individual parts alone. So we're going to need innovative new methods and approaches before we can claim to understand even the simplest living cell. The simplest living cell that we know about is mycoplasma with about 500 genes. We don't understand it, and yet we pretend that we're going to understand humans. And finally, of course, evolutionary pathways provide powerful insights for understanding all of biology. Thank you.
Okay, we have lots of time for questions. Again, I'm going to try my strategy of identifying two speakers, one from each side, taking turns. So I'll pick an extra speaker in advance, so I'm always one ahead of the game, and the persons with the microphones aren't running. So I want to pick two speakers, one on each side. Two questions, one on each side. You might have missed my earlier comment, but I will make an exception for you. You can go first. Okay. But I want to pick a speaker from the other side, as a question from the other side as well.
You may have to repeat the question because I'm a little hard of hearing.
So is there a question from, no, not you, you already asked a question. I want somebody else. You lost me. Student, question from a student, not John. Okay, you can be number two because you've got your hand up. Someone will get you the microphone. Okay, Dr. Davidson.
So I have three comments, aside from the fact that your friends and colleagues really applaud your continuing decision to swim upstream in the discussion of basic and applied research. And
Pardon?
I said your friends and colleagues applaud your decision to swim upstream with respect to translational research and the importance of basic research, and that you mention it all the time, and we appreciate that. But my three comments are the following. One is, what goes around comes around. In 1958, I was an undergraduate senior, and my honors exam question was, quotes, what's the difference between a cell and a bag of enzymes and molecules? Of 1958, it was already, the writing was already in the wall. A second comment I want to make has to do with both your talk and Evelyn's talk, and that has to do with the functional meaning of DNA sequence or RNA sequence, of which not all of it is apparently sequence dependent. Those scaffold link RNAs have rapidly evolving sequence except for the elements of the sequence that serve as docking sites. And so most of that sequence is required to covalently link the docking sites. And I think the same thing is true of a lot of the space and non-coding DNA sequence, which enables molecules to loop, for example. So it's another aspect of the macromolecular structure. And the third thing I want to say is about your example of mathematical models that you gave. In my area of gene systems, far more useful than a computer that grinds away at every possible interaction is the use of mathematics to test and generate predictive outcomes from ideas that are obtained a priori rather than a posteriori.
What's the question?
I think that was three comments and not a question, right?
But one thing about scaffolds I didn't point out is that a scaffold RNA molecule or a scaffold protein molecule has all these flexible sequences. Those sequences will not be conserved. But there should be islands of conservation that bind things in the middle, somewhere along the scaffold. So you don't expect the scaffold. And also, scaffold RNAs and scaffold proteins will generally be large molecules. So these are reasons why they've been ignored mostly. Hardly anybody has actually done the hard work of setting up a vitro system and seeing what they do. And this is just one of many examples why there's so many wonderful opportunities for new scientists, and why many of us are so distressed that young people are being forced into avenues that are already well trodden. Try to solve this disease or do something that is pretty obvious. If you're doing something pretty obvious, there are going to be many people doing it. So, you got one life as a scientist. Try to do something that's different and important.
Okay, John seems to have possession of the microphone. Yeah. But it's not.
There I am. So I'll also reiterate the bravo for basic research. As somebody who failed their comps exam, also failure is good. The stats on Drosophila research for NIH, I didn't realize were so dire, which is interesting because, of course, we even know more about homology than we did when Drosophila research was even more well funded. So what do you see as the path for reminding those in power that work on mice and rats isn't always going to be the best thing, and that these various model organisms, including the protists that you talked about. So what are some solutions?
Well, I'm not sure. My colleagues and I, Harold Varmus, Shirley Tillman, Mark Hirscher, and I published a paper last April on PNAS pointing out many flaws, what we call flaws, structural flaws in modern day biological research. And I talked about some of them. It's just like society, free market capitalism may sound like a good idea, but it always goes wrong and has to be regulated. Free market science, sounds like a good idea, but it has all kinds of ways in which it can go wrong. And so it's in the United States, we're trying to organize groups of people around the country to try to come up with ways of making the way we invest in our young people especially, make it more efficient and more effective for doing good science. My own feeling is that there's too many older people who are happy with the system, that the only way that the system will change is if the young people actually get active. And postdocs in Boston had a meeting with 600 postdocs, wrote a very nice position paper. Postdocs are 30 to 40-year-old scientists, maybe 25 to 35-year-old scientists. It's those people who represent the future, and they have to help structure a system in which will be more effective. And there are many, many issues of the kind you raise that they just don't work right if they're not guided wisely. And we have to do more to make science work better.
Okay, the next speaker is here, but could I first, is there another question? Yes. Somebody who hasn't asked a question yet? Okay. Go ahead.
With our current knowledge in biochemistry, molecular biology techniques, and the like, what can we expect to be able to do in the current next 10 years or so in synthetic biology? For example, apply it in biomedicine or something like that.
So just to clarify for Dr. Alberts, you're asking about the advances that we could expect in the next 10 years
or
in biomedicine, for instance?
Related with synthetic biology.
Synthetic biology.
Yes, because he talked about what we know about biochemistry and the like.
So there are many, many tools now. Synthetic biology is one of them. There's no answer there's no magic wand to figure out what works. We need the judicious use of many different tools. And as I emphasized in talking about my failure as a graduate student, the way I put it is like this. So Rembrandt could paint great pictures. You could give me all the paints that Rembrandt had, and I would make a mess. I could never. So it's the same thing about science. You have to learn how to make all those decisions, that what I called strategy, to do something that's ambitious enough to be meaningful, not so crazy that it will never work, and unique enough so you could make a contribution, and something that's important. I mean, the Barbara McClintock, who I also knew well, used to talk about, and she had this, to do biology, she said you have to have a feeling for the organism. Her organism was corn, maize, and she certainly had a feeling for the organism. I would say to do modern biology, well, you have to have the kind of stuff I'm talking about, you have to have a feeling for the cell. You have to sort of immerse yourself enough to figure out you know, what's really important? What if I solve this mystery, small mystery, maybe small mystery, how that will advance other people's understanding in a broader way, because those are the things that are important. And so what I see often is that physicists, very bright people, come into biology and they start measuring lots of things, but they're often not measuring things that I think are interesting. So it really helps, it's essential really to have a, you know, you don't all have to write textbooks, because that's one way you get it. but you know, you need time to think and read and discuss with your colleagues the nature of the cell, the nature of biological systems, so you could get real good sense of, you know, what kind of problem is meaningful for me to work on that other people don't see clearly, in where I could get a strategy that will make a difference. And that's, you know, that's, you can't do that by just reading books. You have to do a lot of thinking.
My question is about a specific process in the cell, and that is the inheritance of chromatin structures. It's a question about intuition, which goes well with what you just said. How do you imagine this protein machine, if it is going to be a protein machine, to do this task?
So it turns out my hearing aid just died.
So the question is, how are molecular machines, and in particular chromatin structure machines, inherited from one cell division to the next?
Yes. What does your intuition tell you about how a protein machine that permits the inheritance of chromatin structures?
So this is epigenetic inheritance by proteins bound to DNA. This is a very hot field of research. What it needs is in vitro system. You know, you could take that DNA replication system that was just published in Nature, yeast DNA replication system with all the eukaryotic proteins, and actually see what happens. You have to then add purified proteins and see what happens. So right now we're guessing. You know, the guess is that, well, for example, nucleosomes we know remain bound to DNA. At least the H3H4 core are inherited on the daughter strands. I even did an experiment years ago, my postdoc did an experiment with a T4 replication system replicating over DNA with purified nucleosomes on it. And we could show that those nucleosomes didn't leave the DNA. They went randomly to one strand or another behind the fork. And so in principle, the covalent modification of a histone that was there before that histone was replicated will be information inherited by both daughter strands by having an old nucleosomes there. And in principle, something could recognize that modification and spread the same modification again on the new histones, the half of the nucleosomes that are new. But we don't have an in vitro system, as far as I know, unless something I don't know about, that could actually show that that's exactly what happens. And what we do suspect is that some of the modifications, at least in some circumstances, can be inherited. And it's a great, that's the kind of epigenetic inheritance that can cross generations. You know, and but there are some examples in plants where that's clearly happening. So, you know, it's mechanistically we need a lot more biochemistry to figure out what actually is happening in detail.
How can the scientific community work to counteract the mistaken policies of reducing budgets to basic research? How can the scientific community really get involucrated into this task of counteracting this?
This is a question about how can we solve the funding problem? How can we get more resources?
Scientific community counteract this problem?
Well, there's never going to be enough money for science. You know, this is the era of biology. You know, there's so many things that need to be studied. You can't talk about anything in biology without saying there's huge amounts that we need to know. So you can't expect the public to pay for all the science that scientists can do. But what we can do is use the public's money more effectively and you know, apply it to really advancing our knowledge and our understanding in ways that in the long run will be really critical for all the things that the public wants. The public wants you know, to have better ways of preventing disease, better ways of treating diseases. They want to have, you know, we want to have better ways of growing food. We're going to have food problems. We want to have better ways of dealing with the environment. All those things are closely related to understanding biology in a deeper way. So, you know, I think governments have to realize that these investments are critical for the future of their nation, because this is where the future lies, is really innovation, using science to solve the problems that we're generating with ever more crowded world, an ever more warm world, and a place where, you know, as we saw, infectious diseases can travel at the speed of an airplane. So our hope is that science can you know, the advances of science can outrace the problems that we're causing in this world. So governments really have to recognize the importance of science for every society. I've been a science envoy in Indonesia, and even though I'm finished, I'm still going there several times a year. You know, they've way underinvested in science because they don't have the scientific capacity in their nation to solve all these problems that they have as a nation. Nobody's going to fly in from the United States to solve their problems for them. So, you know, how much science is enough? Well, you could look at the standards of government investments in science around the world, and you know, there's been sort of a benchmark set that, you know, X% of gross national product, and so I don't know where Mexico sits in that ranking system, but if you're I'm told it's low. So, anybody here who's controlling funding in Mexico? More money. But again, it's our responsibility as scientists to make sure that money is spent well, and you know, because you know, it's critical that we invest limited resources we have wisely.
One more question over here.
So my question is back on pathways. There is probably another level of complexity when you showed nicely A goes B, B, C, and then there is a positive or negative feedback, is the fact that several of the proteins involved in one pathway can be involved also in other pathways with equal relevance. For example, that's what happens in my field of DNA repair, where we find that protein involved in DNA repair are also involved in chromatin remodeling and transcription factors. So when we study one of these pathways, we have, and we do also mathematical model to understand, we have this additional complexity that the protein could be involved in the other one. And more than being a reason of trouble for the scientist, could be also an important tool to understand how the pathway are connected, and also in terms of evolution. So I wanted just to comment on the fact that situation could be even more complex when you consider that the same protein can be involved equally importantly in different pathways.
Can you repeat this? I'm not hearing it.
So this is just a comment about the importance of the situations where proteins are involved in more than one pathway. And maybe you could comment briefly, how often is it the case that you characterize a protein's role in one pathway only to find that it's also doing something else.
Yeah, I had a slide at the end of my thing about, let me see if I can get this to actually show. I had a bunch of, here. So this is the EGF receptor. Anybody want to make sense of this? Look at this. So everything's connected. So this is an important signaling pathway in mammalian cells. It's involved in cancer. It's involved in development. So-called EGF receptor. And this is a typical network diagram. You know, it's just hopelessly complex. And the answer is that, you know, individual proteins do multiple things. And so how are we going to make sense of this? So I would hope, following the same logic that I use for the brain, that we could come to understand the EGF receptor signaling problem in cancer, for example, by working intensively and trying to see how that process works in worms, how it works in Drosophila, how it works in other organisms. You know, as I said, we have a few model organisms, but we're missing huge numbers of organisms we could actually use to make these kinds of studies. And with our new technologies, it makes it possible to take other kinds of organisms and really find those where a particular problem is particularly appropriate to dissect. You know, so you know, EGF receptor, this signaling process didn't arise all at once. It evolved from, and if we understood its evolutionary pathways, I think we could do a lot more intelligent work to decipher human cells. So, and the answer is really multiple proteins often do multiple things, and we have tools where we can dissect that. I mean, you know, synthetic biology is one place where we can set up systems that are free of other side reactions, and there are many tools that become more powerful every year. And so for a young person to decide what to work on, you have to keep on looking at what are the new tools that we have that might enable me to rethink completely what I'm doing on my research problem. That's an important thing to always do. You know, all of us see a lot of research that just continues down the same track, you know, for decades, multiple decades, and you know, where there are clearly better ways to try to attack the particular problem, but there's so much inertia in our funding systems. You know, if I want to shift, a young person wants to shift and work on Stentor, for example, as Wallace did, most people would never do it because they said, I'll never get resources because I have never worked on this organism. And yet I have a wonderful idea with new tools. That's the only reason I could go back. The only reason why Wallace go back to Stentor is that we have all these new tools that T.H. Morgan didn't have, that Van Starter didn't have. RNAi tools to knock out genes, all these new microscopy tools, you know, engineering gene networks, all kinds of things that can be done now that make Stentor an approachable system for a set of unique problems that can't be studied anywhere else as well. And so we have to figure out ways of allowing talented scientists who have a good idea like that to get resources to do something different. And you know, anyway, my dream would be to have a funding system for starting independent scientists, which is like a pathway we have a small amount of money for in the United States. It's called a New Innovator Award, and for that award you have to be less than 7 years after your PhD degree. You have to propose something that's innovative and risky, and you don't have to have preliminary results. But what you do have to have is a track record that shows you could do science. So anyway, some system that encourages young people to jump onto really important problems with a new approach, rather than feeling that they're forced to continue doing the same science that their advisor did in order to get supported. That's the critical thing that needs to be emphasized, in my opinion.
Okay, I think we'll stop there. We should thank Dr. Alberts and Dr. Keller, and we have, I think, a half-hour break.
I have three things to tell you. First, there has been a switch in the program. Tomorrow we will have Dr. Davidson talk at 9 a.m. instead of Dr. John Loxton talk, and Dr. John Loxton talk will be at the place of Dr. Davidson on Wednesday. Also, I invite you to take a look at our souvenirs. These funds, the funds collected, will help to fund the meeting, and the coffee break is open, and we have a half an hour break. Thank you.